Good evening, sir. I'm fine. How are you?
I'm good. I'm doing good. Okay, so in last class, we figured out how DNA is called happens to be the genetic material and how the scientific community was convinced. So all of you are convinced now based on evidences that indeed DNA is the genetic material and DNA is the one that you know is responsible for inheritance or whatever Mendel figured out without even knowing anything about DNA now. So are you all convinced? We have Iram joining in as well. Good evening, Iram. How are you doing?
Yes, sir. Good evening. I'm good. How are you?
I'm good. I'm good. And for your knowledge, the spotlight feature is on. Okay. Okay. So everyone is convinced that DNA is the genetic material and how we reached that stage in science. How did scientists figured out that indeed it's DNA? Anyone has any doubts, any confusion till now? If everything is clear to everyone, give me a thumbs up and we will move forward today. Okay, cool. So everyone is quite clear. So let me start a new page just for some simplicity. And let's start here. So basically, we also learned about RNA world hypothesis. We also figured out that before DNA, it was RNA. And the evidence is that at one point of time, back in the biological past, RNA was acting both as a genetic material and also doing other jobs like of a catalyst or also to scaffold. Some scaffolding roles were also there, and also other processes like metabolism, translation also evolved around RNA. So far, so good. But now let's come back and discuss more about the theme of central dogma that is going to be the most factual part of the chapter starts now. And but the good thing is that these are mechanisms, so they can be understood and they can be memorized by some inherent logic in the process. So the first thing, let's start with this stroke. This one. So the first thing that we'll begin with is replication of DNA. Now, who figured out the double helix structure of DNA? Quickly, just a revision.
Watson and Crick.
Perfect. So the double helix structure was given by Watson, J.D. Watson, and Francis Crick. Okay. Now, Watson and Crick did this in around 1953. Yes. And the moment they realized that DNA is a double helix, intuitively, this question also came to their head that if DNA is a double helix molecule, it makes all the sense to think how it replicates and how it propagates. So when one cell divides to form two cells, this DNA needs to be replicated and then it needs to be shared equally between the daughter cells so that both the daughter cells have the same genetic material that the mother cell had. Now, one way to test this is in the simplest of the organisms, which are bacteria. So when they found the structure, they also hypothesized a mechanism by which DNA replicates. Okay, so they hypothesized at that point of time. This was not tested. It was just a theory hypothesized by Watson and Crick based on logical explanation of how double helical structure can replicate. Now, think about it. In biology, if something is a double helical structure, right? Something is like this, and we know that it replicates, and it has bases projecting from both the sides. Bound together by hydrogen bonding. Now, logically, how will you think it can replicate? This is what Watson and Crick also did. What is the way to replicate, like to make its copy? So they suggested that there are two strands, right? You see, this is strand 1 and this is strand 2, correct? And they are like a zipper, which is zipped from the center here through hydrogen bonding, right? Do you understand?
Yes, sir.
And hydrogen bonding can be broken. Hydrogen bonds do break both through enzyme activity, like which is chemical activity, can also break by heat. So it makes it easier for a DNA to just unzip itself and open into two strands separately by breaking the hydrogen bonds which are present in the center. And then once they separate, these two strands then form complementary strand. Okay. So his idea was that these two strands open like this. Okay. And when they open up, now they look like this because they have opened from the center. So the hydrogen bond between the nitrogenous bases have been broken. And what happens is that they form a complementary strand around it. Not like, and complementary, when I say complementary, I mean the complementary base pairs are there. And then later on, you can just, what do you say, coil it in a right-handed fashion, and it again becomes a double helix structure. So first, for replication, the double helix structure needs to be uncoiled or unwound, and then it needs to open, separate apart, and then new complementary strand will be formed. So in this case, we call this as the template strand, template, and this becomes the new strand. Okay, do you understand? We also call it, let's call it parental for now, because template is actually one of the strand. This is parental strand, and this is new strand. In this case, this is parental strand, and this one is new strand. Do you understand what they proposed? Do you understand the theory that they proposed? That this is how DNA, from one DNA makes two copies. Okay, is that clear, everyone?
Yes,
Yes.
sir.
And they proposed this as, this model was called semi-conservative model of DNA replication. Why it is called semi-conservative? Can anyone tell me? Just look at here. Can you say, tell me why it is called semi-conservative? What do we mean by conservation or conservative? Conservation means to keep something which is, like to preserve something which is old, right? When we conserve forest, when we conserve wildlife, we call it a conservation project for things, right? This term is also used in social aspects when we call a conservative society, where the society has just preserved the old ethics, values, and beliefs and does not update with time. So we call it conservative thought process or conservative societal setup. So conservative means whatever when old things are preserved. Semi means half, semicircle, right? So semi-conservative means whenever a DNA replicates, half of the DNA is the older one only. So half of the DNA is old, which is conserved, and the new half is synthesized around the old half. So it's called semi-conservative. Does that make sense, everyone?
Yes,
Yes,
sir.
sir.
Yeah. So let's write down about this scheme. Write down. Watson and Crick proposed. Watson and Crick proposed the semi-conservative scheme or model of DNA replication. This model suggests that the two strands of DNA will separate and act as a template for the synthesis of new complementary strands. Does that make sense, everyone?
Yes.
Yes, sir.
Now, pay attention here. So there are some details that you might like to understand. So this was strand 1. Let's say where it is going. If I follow, so this is strand 1. Okay. This is strand 1, original. And this black one is strand 2, original from here, right? Now, if you see this complementary strand, which is the new strand, let's call it 1 prime. And let's call this one as 2 prime. We are just naming it. Now, 1 prime, the sequence of 1 prime will exactly be same as which strand? 1, 2, or 2 prime. There are 3 options. The sequence of 1 prime, which is a new strand formed complementary to strand 1. So this strand sequence will exactly be same to which other strand? 1, 2, or 2 prime?
2.
2. Who says this?
Alia.
Okay, Alia says 2. Iram, what do you think? You're also unmuted.
2 prime.
2 prime.
So which strand? 1 or 2? Sorry, 1 or 1 prime?
1 prime, 1 prime. So this newly formed strand, 1 prime, the sequence of 1' will be exactly same to which other strand, 1, 2, or 2'? Rabia? Iram? What do you think? Alia says 2, which is this one, the original strand 2. Anyone else? Any other answers? Come on, quickly. It's not that difficult a question. So if you see, go back. If you go back like this one was bound or was complementary paired to 2. Okay. Now, when you opened both, you again make a new strand. So again, 1 will be complementarily paired to something which is 2 only, right? So new strand 1' will exactly be like the old strand 2. And the new strand 2' will exactly be the same like old strand 1. Does that make sense, everyone? So this strand will be like this, and this strand will be like this. That's why they are complementary. That's why a new DNA is a new, 2 new molecules are formed. Is it clear to everyone or not? People, yes, no?
Yes, sir.
Yeah. Okay, let's simplify it a bit. Let's say the parental strand here, this one. Let's first talk about these 2 strands. So let's say strand 1 has the following sequence: G, C, T, A, T, A, C. So what do you think strand 2 sequence will be here in the real ones, in this case? Tell me, quickly, quickly. This is before replication. Strand 1 is G, C, T, A, T, A, C. So strand 2 will be? Parker? G always forms bond with C. A forms bonds with T, AT, and G forms bond with C. So the sequence in the second strand will be C, G, A, T, A, T, G, right? Yes, Aisha, correct. Why are you not speaking, Aisha? You should speak. So this was the double helix, okay? Now what happened after this separated? So when this separated, again, the complementary to this sequence will be this sequence only, right? So now you can understand when 1 got separated from 2, then the new strand which will synthesize will exactly be complementary. Okay, when 2 will get separated, it will again get a same strand. Otherwise, they cannot bond together. Is that clear to all of you? Yes, no, people?
Yes, sir.
Yeah. So the sequence remains same. It's just that half of the DNA is newly synthesized. Okay. Now, but this is all theory. This is all a story that proposed, not, I should not say story, but a hypothesis. In science, before we start to prove anything, we should need a hypothesis. We should say what we expect or what should be the outcome of an experiment. Then you do the experiment or you find a way to do an experiment to either prove or disprove the hypothesis. Okay. So this was a hypothesis that DNA must be opening into 2 parts and half of the strand will be newly synthesized. But this is just a hypothesis. And we cannot believe just because Francis and Crick said this. Even whoever said this, regardless of who said what, we need proofs. So where do we get the proof from? So we got the proof sometime later. I think it took around 5 years for the proof to come. And when it came through a very, very beautifully designed experiment, which is still till date is known as the most beautiful experiment in biology. It's regarded as the most beautiful, clear experiment in biology. Now, what was the experiment? The experiment came from Messelson and Stahl. It's known as Messelson and Stahl's experiment. So let's go to see what was the experimental proof. So remember, someone else proposed, but someone else proved it. Experimental proof. So read the question carefully. If they're asking you who proved the semi-conservative nature of DNA replication, then you don't have to talk about Watson Crick. You have to talk about Messelson and Stahl's experiment, which we are going to do now. If they ask you who proposed, then you have to talk about Watson and Crick, because they proposed. The moment they figured out the structure, this was the most logical thing that came to their head. And if you will read their original manuscript, they have written there in back in 53, when they figured out the double helix structure, they said that it has not escaped our attention that the double helix structure and the pairing that happens in the double helix structures immediately suggest a possible copying mechanism, a mechanism to copy DNA information as the genetic material. So this is what they have also written. So that's where they propose that this is a very, very logically possible way through which it must be happening. But experimental proof for semi-conservative model came in 1958. And there were two scientists called Messelson and STAHL, Stahl. So Messelson and Stahl's experiment. And this is very, very beautiful experiment because it is so simple, everyone can understand it. And it also proves that, yeah, it is happening. Whatever is happening because of semi-conservative method. So you all can see the screen for now. I'll use a red ink. I should use blue ink because blue is not here. Okay, so what they did was very simple. We know that DNA has a nitrogenous base, right? And to do experiments in biology, you need to use a system, a model system or an artificial system, which you create and you test things in that. So they used E. coli bacteria. E. coli was the model organism, okay, in which they tested this. Now, the reason they chose E. coli was, and you can write it down, because it's a prokaryote and it has just one single chromosome, which also means that it has just one single thread of DNA, right? This thing is clear to all of us. Now, it's easy to work with single DNA because you can very easily quantitate. Now, what they did, they took E. coli bacteria and they started culturing the bacteria in a medium. You know, you need medium to culture bacteria. And in that medium should be the nutrition for the bacteria so that bacteria can do their DNA replication, cell division, and all the processes, etc. So they also cultured the bacteria, but they used ammonium chloride to culture bacteria, okay? Ammonium chloride is NH3, sorry, NH4Cl, 5O. It's 5, no? The valency of nitrogen is 5, right? So 4 taken by hydrogen, 1 Cl. So ammonium chloride. So they used ammonium chloride, but they used a heavy isotope of nitrogen. Okay. So they use nitrogen 15, N15. What is the atomic number of nitrogen? Generally, that you read in periodic table, what is the atomic number of nitrogen?
Seven.
Sorry?
7.
7. What does that mean? If the atomic number is 7, what does it mean? What decides atomic number? Come on.
Number of
chemistry.
electrons.
No. If you write this down in any exam, any high-end exam, you are gone. Atomic
group
number
number.
is no. Atomic number is decided by number of protons. Number of electrons can change when you make ions. Number of electrons add or subtract. If it's a positive ion, it has less electron than protons. If it's a negative ion, it has more electrons than proton. But protons stay the same. Number of protons do not change under normal circumstances. So it's the number of proton that decides atomic number, not the number of electron. Normally, when you read periodic table, it looks like nitrogen has 7 electrons, 7 protons. So it must be the number of electrons that decide atomic number, but that's not true. Okay, always remember it's number of proton. So 7 protons are there. That's why atomic number of nitrogen is 7. What is the atomic mass of nitrogen?
About 14.
14,
14.
right? And why is that 14? Where is this mass? What is atomic mass decided by?
By electrons plus protons.
Do electrons have mass? Come on, people, you have to buck it up. You have to start revising. Electrons do not have mass. It's good that you're making all these mistakes so that you will learn. If you would not have made mistake, if I would not have asked you this question, you would have just thought the wrong notions in head. Electrons do not have unit mass, right? They are called massless, right? So Alia is writing protons and neutrons, and that's the correct answer. The atomic mass of an atom is decided by neutrons and protons. And what are neutrons, Alia? And where are they?
They are neutral and they are in nucleus. Yeah.
What do you mean when you say they are neutral? Yes, they are neutral. That's
don't-
why they're called neutrons. Yes. What
Yeah,
do you mean
they
by
have no charge.
they do
Like
not have any charge? Like
protons
protons,
are positive and electrons are negative. Neutrons have
right?
no charge. So they are
Right.
neutrons.
But what do they have?
Yeah, that I don't know. Yeah.
Yeah, they have mass. That's what you're
Okay,
saying
yeah.
now.
Okay, yes, okay,
Right?
yeah, yeah.
They have mass, but they do not have a charge.
Yes, sir.
Electrons have a charge, but they do not have a mass. They do have mass, but it is so negligible in comparison to proton and neutron that we do not consider it. It's like an elephant and an ant, so you don't consider it, right? So electrons are called massless mostly. So they cannot contribute to the mass of an atomic mass. So the mass comes from number of protons plus number of neutrons. And both protons and neutrons are present in the nucleus. Hence, they both are called nucleons. So nucleons are present in nucleus, and that's where mass comes from. So almost all the mass of an atom is in the nucleus. Electrons that circle around in in discrete orbits do not have a mass, but they do have a charge, which is negative. Okay. In a normal neutral atom, the charges are balanced. The number of protons in the nucleus is equal to the number of electrons revolving around. So the charge is balanced and the atom is not charged. Okay. If you want to make an atom into an ion, that we read, for example, Cl negative, chloride negative, or H positive, you just have to either remove an electron or add an electron in the orbit. You cannot remove protons from the nucleus. It is very, 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 very difficult. And it's called atomic fission. And atomic fission, it will breaking down the nucleus will it will need so much of energy and it will produce so much of energy that it's not doable normally. But taking out an electron is very easy. In reactions, normally we do that. So normally the mass of nitrogen, normal nitrogen, should be 14, right? N14. Yes or no? People, are you with me? If anyone slept, just let me know. Are you all, because I cannot see you, so I do not understand if you are with me on the same page, because these are fundamental. These are very important. Yes, Alia is with me. Oh, no. Parker is with me. Okay. So normally nitrogen's mass is, atomic mass is 14, but there is a heavy isotope of nitrogen, which is 15. So its mass is 15 units. Now, can anyone tell me how can a nitrogen have 15 unit mass? What changed here? It has 7 neutrons, 7 protons, and 7 electrons under normal condition. What can you change to make N14 into N15? Okay. Can we change electrons and make a N14 into N15? Parker says electrons. And we just discussed, so Parker is making the classical mistake of not paying attention to the details. So remember one thing, a very important person and a very important teacher in my life always tells me that both devil and divine lies in details. Okay, so you have to pick. You have to also pick, you can also pick divine from details, you can also pick devil from details. And mostly we do not pay attention to details, and that's where devil takes our marks away. Okay. By removing or adding electrons, we cannot change the mass of an atom. Do you understand that, Parker? Here we are changing the mass. It should be 14, but we are creating a 15 atomic mass nitrogen. So it is a heavier nitrogen. It is heavy. So, and you're saying if I change electrons, N14 will become N15. That cannot happen. So what we need to change? Come on. Someone has to come up with a concept. These are very, very important and simple concepts. And I think you studied, when was first atomic periodic table was introduced to you? Wasn't it in class 10th? 10th, right?
9.
9th. Okay, great. So 9. So it's been 4 years now. 4th year you are in, or if it was in 10th, you are in the 10th and 11th.
The 9th was online.
Sorry? 9th was online. Yeah, that's so, COVID did not just take health from human civilization. It also took a lot of knowledge. Students who did anything in the COVID, whether it was schooling, graduation, post-graduation, they could not do what they should have done. So I completely empathize with you all. But that cannot be an excuse. So remember, no one is going to give you grace marks because you could not study 9th or 10th because of COVID. They're still going to judge you. The world is brutal. Everyone forgot about COVID already. So when you will give your exam or you will sit for entrances, no bounty points for suffering, struggling, and emerging victorious through a pandemic will be given to anyone. Okay, do you understand that? So let's keep it aside and come back to brushing up basics. So what can we change? We cannot change electron. What can we change to make N14 into N15? Rabia? See, science is all connected. If someone joins now, they will think that why is this teacher teaching chemistry using biology on the board? There are only 2 candidates left. Come on, make a guess.
I think neutrons.
Alia
Because
thinks because
you said the protons are hard to remove. I think maybe I misheard. So maybe
no,
maybe
you're right. But just because I did not say that neutrons are also hard to remove,
okay.
that doesn't mean that neutrons are easy to remove. Both proton and neutrons are very hard to remove from the nucleus.
Okay.
But they can be added.
Yeah, I mean like one is added. So because it's 15, so 14, so one is added.
So why do you think it's neutron? Why can't we add just one proton?
Like I said, because
I really appreciate that you are discussing this with me, Alia. You may not be understanding, but this is the real essence of education. Yes, come on.
I thought that since protons are hard to remove, maybe they are hard to add on. So yeah, so neutrons. So yeah, okay.
Both protons
Maybe
and
it's
neutrons
proton.
can be added, let's say. Let's
Okay.
assume. Now, given this information that you can add both, what will you add to increase the mass of an atom, of an element?
Okay, proton? No,
Proton.
neutron.
Okay, now you proton.
Yeah,
Okay,
proton.
so you change your answer to proton. Okay, that's also very good. But remember that the number of protons also define another criteria for an element, which is atomic number. How many elements can have same atomic number?
None.
I am sure that in class 10th, you must have studied about isotopes. Right, Parker? Parker says yes, but what yes? It's like in schools, when I was in class and when I did not, when I used to not know any answer, what I used to do is I used to just look around that what people, the mass is moving their head like. So the teacher asks, so do you all understand that proton defines atomic number? I will just look around and everyone is like doing this. So I'll do yes, I will also do this. And then in that crowd, no one will know that I have not understood anything. But for a teacher, it's like, oh, wow, everyone understood. Everyone is shaking their head like a group of lambs. Right? So Parker, what yes? If we change atomic number, what happens? Simple question, simple fundamental question of chemistry. If I change the atomic number, what happens? If I go from atomic number 7 to atomic number 8 by adding one proton, what happens? Come on, people, what happens? You move ahead in periodic table. No, the reactivity decreases or increases is correct, but the whole element changes. If it is atomic number 7, it is nitrogen. If it is 8, it is oxygen. You cannot have a nitrogen with the atomic number 8, same as of oxygen. One element can only have one atomic number. That is how it is defined. 1 is hydrogen, 2 is helium, 3 is lithium, 4 is beryllium, 5 is boron, right? 6 is carbon, 7 is nitrogen, 8 is oxygen. That's how you remembered periodic table, or you do not know anything about periodic table? Yes. So if I go from atomic number 7 to atomic number 8, which is defined by the number of protons, I'm changing the element. That's why we can't do that. We cannot take nitrogen, add a proton and make it oxygen. Let me tell you. If this was possible, the whole world would have been taking anything that is around, adding up protons and making everything in gold. You know, we would have no water, no iron, no oxygen, no nitrogen. We don't need these things. We just need gold. So club all the protons, put them together, make gold. Gold is also an element, right? But good that it cannot be done. That's why nature is at balance. So we cannot add proton to make nitrogen heavy, because if we add proton, it will become oxygen theoretically. So we can only add neutron, right? So adding neutron does not change atomic number. Therefore, the element remains whatever it is, but it changes atomic mass. So the element becomes heavy. And these elements are called isotopes. So a nitrogen can have two different isotopes, a heavier isotope or a lighter isotope. N13 can also be there. N15 can also be there. N14 is normal. But all three are N, nitrogen. They all have 7 protons. So all their atomic numbers are same. Their masses are different. Is this clear to all of you?
Yes,
Give me
sir.
thumbs up. Give me 2 thumbs up for teaching chemistry and biology both at the same time. This is also a proof that everything is connected. So what Messelson and Stahl did, they very beautifully decided that let's do one thing. Let's use a heavy nitrogen, N15, and culture E. coli bacteria in a media where the only source of nitrogen is this ammonium chloride with a heavy nitrogen in it. So you can synthesize these chemicals in lab, and then you can put this on the culture plate with no other source of nitrogen available. Now, if there is no other source of nitrogen available, what will happen? Let's go with generation 1. Sorry, parent generation. So this is the parental generation, the parent bacteria. So normally, sorry, 1 second. Yeah. So this is the cultured bacteria, which now acts as the parent. So if you culture a bacteria on heavy nitrogen, every 20 minutes bacteria divide, right? 25 minutes around, bacteria divides. E. coli we are talking about. So if E. coli divides every 25 minutes, it will need a lot of nitrogen to make new DNA, to make other things in the cell. Yes or no? Everyone understands? And the new DNA is only the heavy, sorry, the new nitrogen is only the heavy nitrogen available. So after many generations, after 2, 3, 4 weeks, if that culture is still there, you will know that all the bacteria now which are present on the plate are product of divisions, and they all must be having nitrogen 15 in their DNA, right? All their nitrogenous bases now contain N15, not N14. When you place them in the beginning, few bacteria, they were having N14 in their DNA. But as time progresses, bacteria divides. Over time, they use new nitrogen from the media, and the new nitrogen is a heavy nitrogen. So all the bacteria will be now N15 labeled. So they will be heavy as compared to normal bacteria. So this is the parent, which is the cultured parent on ammonium chloride with a heavy nitrogen in it. And after 20 minutes, when generation 1 comes, now you took these bacteria, which were growing on heavy nitrogen, and you introduced it to normal nitrogen. So what will happen? When you put it to normal nitrogen, after 20 minutes, when cell will divide, it will again use nitrogen. But now it does not have any heavy nitrogen available. So when the new strand will be formed, it will only contain N14. in the DNA. And if Watson and Crick's proposition was true, if their hypothesis is true, one strand will still have, because see the parent, both the strands are red, which means both the strands were heavy. Red is heavy, green is light. So here we started with heavy parents. After 20 minutes, these two strands opened up, and the new strand which is formed will be of N14. The green strand is N14, lighter or normal nitrogen. Does it make sense, everyone? Yes or no? Don't look what's at the bottom, only look the front, the top part. Is it clear? Then thumbs up. If not clear,
Yes.
okay. So after one generation, we will have a hybrid DNA. One strand heavy, one strand light, one strand heavy, one strand light in the progenies. After 40 minutes, which is like when the second division will happen in normal nitrogen. Okay, when I say normal nitrogen, I mean N14. Then again, these two will open up. Now from 2, we will make 4. So when they will open up, there will be one green and one red, one green and one red. But all the complementary strands will be green. They will not be green. We are just representing here as green. All the complementary strands will be light because only normal nitrogen is available again. So after 40 minutes, you will get 2 strands which are completely light strands, whereas 2 strands will still be hybrid. Do you understand how this happened? Just look at the diagram and try to see if you understand. People, everyone, pay attention and see if it makes sense. I will take a quick water break and I'll be back. Just look at it and see if it makes sense to you. Is it okay, everyone?
Yes.
Okay. Everyone, does it make sense? What's on the board? Yes, no? Anyone who's confused, just speak up or just write in the chat that you want me to repeat whatever I said. Okay. So they saw that indeed what happens now, if this is happening in reality, how will you get to see it? You cannot see bacteria. You cannot see DNA for sure. Forget about seeing a red and a green strand. These are all representatives images. how will you see it? And that this is why this experiment is called the most beautiful experiment, because it's very, very fundamentally simple. The reason they chose a heavy isotope is that you can use the force of gravity through centrifugation. Remember, a similar thing was done by was done by Hershey and Chase, right? Alfred Hershey and Martha Chase. To prove that DNA is the genetic material, they also centrifuged. But there it was the centrifugation between viruses and bacteria. Bacteria being heavier settled down first. Here it is between two different kinds of bacteria. One which contains heavy DNA inside and one which contains light DNA. So the heavier DNA will settle first. You will get a band. So this is a tube which is joint here, and then you just spin it. So imagine like you going on a round. What are those swings called where you sit? The seat is tied by a rope and then everything goes. Huh? Merry-go-round. Merry-go-round goes like this. I'm talking about this. Suppose you tie. So I take this bottle and I tie a knot here and I start moving it. So the pressure of the force of water will move towards outside, right? You understand? When I'm doing this, do you understand what I'm saying? So
Yes.
imagine this bottle as a test tube. In this test tube, there are two different kinds of bacteria growing. And then in a liquid media, one has heavier nitrogen in their DNA, one has lighter nitrogen. And then when I spin it in a horizontal manner, not like, I'm not saying vertical. Vertical will also work the same, but they did it horizontal. Then the heavier thing will start settling at towards the base first, right? So you will see a heavy DNA, which is N15, N15 on both the strands, will make a band here. Now, again, these bands also won't be different colored, but they will be different position. So this is farthest from the center, from the cap. Then a DNA which is hybrid will make a band somewhere in the, between the heavier should have come here. So you see the distance reduced. And if there are lighter DNA strands which contain both the strands having lighter nitrogen, N14, N14, then it will make a band, even less distance from the cap, okay? From the beginning. Do you understand? Heavier things move towards the boundary when you centrifuge them. Is it correct? Do you understand? That's why when kids are on, on those playground jhulas, what do you say that? Swings which go like this. I think it's also called merry-go-round, a horizontal merry-go-round. There, if you see a small kid, which is towards the center, versus a big kid, which is towards the center, at the same speed, the big kid will feel more force to go towards the end, right? Because simply it is heavier and the force of centrifugation is working more on it. Is it clear, everyone? So what they saw when they started the culture, they took one culture and did centrifugation. They found only one band, and that band was also towards the very end of the tube. Now, if you find only one band and that is towards the very end of the tube, what does it tell you? It tells you that all the bacteria contained heavy DNA, purely heavy DNA, N15, N15 on both the strands in every nitrogenous base. If you find a single band again, but not towards the end, but towards the middle, then you can say that maybe it was a hybrid one because it had one heavier strand and one lighter strand. So it means hybrid. So after one generation, they found only single band and the band shifted towards the middle, which proved that yes, what Watson and Crick said is correct. DNA indeed replicated in a semi-conservative method. If DNA would not have replicated in a semi-conservative method, then what would have happened here? This is case A. Then after one generation B, and then one generation C. Let's say what Watson and Crick said was wrong. Let's assume, let's do a thought experiment. So if they were wrong, what would have been difference in the result? Anyone? This will prove that you have understood this. Let's start with Mariam. Mariam Khatun. Mariam, are you there? Can you speak up?
Yes, sir.
Yes. So Mariam, my question is, if what Watson and Crick said or hypothesized was wrong, let's assume, in mathematics, we always say, let the value of X be this. So let's assume it was wrong and DNA instead of semi-conservative method followed a fully conservative method, which means the older two strands stay together and two new strands are formed together. In that case, what would have been the difference in the result section, in the ABC you would have seen? Alia? Mariam, do you understand or you? Yes, Alia.
Okay. A and A is a parent, so it would be same, right?
A
And
would have been same, perfect, because in A we are not doing anything. Yeah.
yes. And I'm not very sure about B, but I think in C, there's the hybrid, right? So hybrid wouldn't be like, it would be um N15 with together, and there will be N14. Like there won't be a hybrid. I'm not very sure about B.
Okay, so you are saying that you don't know what will happen at B, but at C, you will,
Yes.
how many bands you would have seen if semi-conservative method was not followed and fully conservative method was followed by DNA?
I think three, or
Three.
no. Okay. So one was like 1, 2, 3, 4. Okay, 4. But it will be like the hybrid will be like M15, and there will be M14. So 3 M14 and 1 M15. I think. Yeah, maybe.
3 and 14 and 1 and 15.
Yeah, I think. I'm
Okay,
not very sure.
3 and 1, but it's a good try. I think what you said before made more sense to me. You said that the hybrid band will not be there at all in C.
Yeah, there will. Yeah, like there will be no hybrid band. I didn't say that there
So
will be.
how can 4 bands be there? At max, there can be 3 bands. I'm surprised that you say that there are 4 bands. See, the 3 bands could be lighter, hybrid, heavier. Lighter at the closest to the cap, hybrid in the middle, and heavier at the bottom.
Yeah.
Okay,
Okay.
towards the end. So at max, there can be 3 bands. So will we see all the 3 bands in that case, or we will see just 1 band or just 2 bands? We cannot see 4 bands at all. What is the 4th band?
Okay.
Nothing.
So I mean,
Come
I
on, try
just
again. Yes.
use.
Yeah.
Okay. Um.
Okay, do not panic. Let's do one thing. Let's look at parent. This is important,
Yeah.
people. If whatever method would have been followed, parent would be unaffected, right? Because it all starts from the parent. So when you do the same experiment, and let's say that Watson and Crick were wrong, then for parent generation, you will still get the same centrifuge result, right? You will only get one band, which is heavy DNA, because all parents have heavy DNA, because all of them have been cultured in heavy nitrogen medium. Is that clear to everyone? First step is clear? Perfect. Now, in this experiment, what they found is that after one generation, they are still getting one single band only, but the band's location shifted. The band is not towards the bottom, but it is towards the middle, which tells us that it was not, the DNA was not as heavy as it was in the parents. And there is only one way to make a DNA less heavier. See, one strand is already heavy in the parent. So in the parent, both the strands are heavy. There is only one way, that you open both the heavy strands and you make two lighter strands, because now the parents are being cultured in a normal N14 medium. So the fact that you see a single band and it has shifted from bottom towards the middle proves that the mass of DNA is reduced, which means both the strands are not heavy anymore. But since it has not shifted so much that we can say that both the strands are lighter. So both the strands are hybrid. That's why we just get one band. What would have happened if instead of semi-conservative method, a fully conservative method was followed? In that case, these two strands would have stayed together. That's what fully conservation. So two red strands or two heavy strands would have stayed together. And the two new strands which were formed would have been both light DNA together, right? Because now the parents are being grown in N14. Is that clear, everyone? These are the questions that come in entrance. That's what I'm preparing you for. So you would have seen two bands. My voice is cracking for Parker. She's saying, is my voice cracking for Alia, Iram, Mariam as well? And Aisha?
No, sir. No, sir.
No, sir.
Is it clear? Okay, so I think Parker, then it's your internet. Please check about it and let me know. So, okay, do you understand, people? If semi-conservative method was not followed, then the parent strand, even if they would have opened, it would have come again close together so that they are conserved completely. And the new daughter DNA would be completely light. Yes, Parker, you can rejoin. And instead of getting one band, you would have gotten a band like this. So in the tube, you would have gotten two bands. And this time, no hybrid band, one band which is heavier towards the end, and one band which is lighter towards the beginning. But no hybrid. The hybrid band proves that two heavy DNA strands opened and the new light strand synthesized complementary to both the heavy strands. Does it make sense, people?
Yes, sir.
Everyone?
Yes.
So in NEET, what was asked is, let's assume that DNA replicates in a fully conservative manner. Then in the generation 1, how many heavy, hybrid, and light DNA you will find? So remember, when they say generation 1, it means only 2 DNA will be there because one bacteria divides into 2. So how many heavy, how many light, and how many hybrid will be there if fully conservative method was the true one? Iram.
One heavy and two will be light.
One heavy and 2 will be something, which means 3. Do you see 3 DNAs in generation 1? How many
No.
DNA do you see in generation 1?
One.
Sorry? How many DNA
One
do you see in
DNA.
gen? 1. You see 2. I can see 2. See? Let's count. 1, 2.
Yeah.
I can see 2 DNA, right? I used to watch Oswald when I was a kid. And the one thing I used to like about Oswald is, anyone has seen Oswald as a cartoon? Anyone? Just raise your hand if you've seen Oswald. So what Oswald will do is, every morning when he will have tea, he will put marshmallows in it. And he every time will have 3 marshmallows, but he will count 1, 2, 3. And it was so soothing to, yes, Alia, you must be knowing that, right, Alia?
Yes, sir.
So we just counted that in generation 1 bacteria divides to form 2 bacteria. And 1 bacteria has only 1 DNA, which is 1 chromosome. So 2 bacteria will have 2 DNA or 2 chromosomes. So how many of the 2 will be heavy, hybrid, and light if fully conservative or conservative method was followed and not semi-conservative? Yes, Iram, now you answer. The answer is already on the screen.
One will be heavy and one will be light.
Exactly. One will be heavy, which came from the parent as it is conserved. One will be light, which formed during division, cell division, from N14 normal nitrogen, which is now present in the environment. So 1 heavy, 1 light, no hybrid, 0 hybrid. But in fact, they got no heavy, no light, 2 hybrids, which means all are hybrids. Does this prove that DNA indeed replicates in a semi-conservative manner? People, do you all
Yes.
understand? Everyone understands this experiment? Yes, no? Okay, now let's go to generation 2. And the answer will be given by Alia. So, or Rabia. Rabia is not. Rabia, are you there? Rabia, can you speak? I think, yes, Rabia. So can you speak up? I think no. Aisha, can you speak up? Whoever speaks learns more, because I'm going to ask the next question to them. Aisha, not Parker, but Aisha. Aisha. Aisha is okay. Aisha is traveling and she's joining class. That's amazing in itself. So let's not. Okay, let me go to Alia then. Alia, you can speak. Okay.
Okay.
Okay, Alia. So my question to you is, if let's assume DNA replication happened in a fully conservative manner, then in generation 2, how many light, hybrid, and heavy strands you will get? So when I say generation 2, first you have to understand that total strands in generation 2 will be 4. So how many light? Yeah.
2 will be light and 2 will be heavy.
2 will be light and 2
2 will
will
be
be heavy.
heavy. Yeah.
Okay. Is Alia correct, Iram? Do you think she's
No.
no?
No.
No. Iram says no, Alia won't be correct.
No.
So what do you think, Iram? What is the right answer? Or let go back to Alia. So Iram says it won't be correct. So Alia, would you like to change anything in your answer?
No, because I think like 2 light, because or maybe not. Okay. Okay, I'm getting confused now.
Exactly. Concepts come from confusion.
Yeah.
See, the first 3 alphabets of confusion and concept is same. So we all start CON, and then if you pay attention, you can make CEPT, or if you don't pay attention, you FUSION. So either from con, you can go to concept or confusion. So it starts same. So confusion in the beginning is a very good sign that you are learning. If you are confused, then only you can learn. But if you're constantly confused and confused till the end, then you're confused.
So
Yes.
like in generation 1, right?
Why
So there
don't
are
you
two.
do? Let's do one thing. Let's do one thing, everyone. Alia, do you have two colors pens in your hand right now?
I had.
No.
Wait a minute. Yes, yes.
Yes, two color, black, blue, any two color, or pencil and a pen. Anything do you have? Two different
Yeah,
colors.
yeah.
Why don't you draw yourself? Everyone, let's draw, starting with a heavy DNA in the parent and assuming that instead of semi-conservative method, it is fully conservative method. Please draw generation 1, 2, and also generation 3, which is not shown here. Quickly, everyone. It's fully conservative method, not semi-conservative, which is the correct thing that happens in biology. And you have to tell me how many light, hybrid, and heavy strands you will find in generation 2 and 3, according to that, if that was true. You have 2 minutes, everyone. Come on, draw it.
You're done.
Who's done? Who's done? Who's done?
Me.
Iram is done. Okay. Alia, what about you? The confusion
I'm
started
done,
with
sir.
Alia.
I'm
Done.
done,
Okay,
sir.
perfect. Alia is done. Mariam, Parker, Rabia, and Aisha. 4 of you. Aisha, I think, is traveling, so I don't know if she could draw it, but maybe if you could do the thought, if you could draw in your head, just close your eyes and try to imagine. Rabia also is, I think she's saying she's out. It's great that you people are joining classes, even if you are out of your houses, but yeah, Aisha and Mariam, oh, sorry, Parker and Mariam. What about you? Done? Parker says done. Mariam Khatun. Done. Okay. So I want all 4 of you to write answers in the chat, even all 6 of you. If Aisha and Rabia have done it in their mind, please write the answer. Write like this. Use the word like LH or LHB and H. HB for hybrid. So L is equal to number, HB is equal to number, H is equal to number in generation 2 and generation 3. Come on, send me your answers.
Can you please repeat that? What you said?
The question is,
Yeah.
considering or assuming that DNA replication takes place through.
No. How do we write in the chart?
Yeah, write in the chat like Gen 1, Gen 2, generation 2. And in front of that, L, which is light, is equal to X. HB, which is hybrid, is equal to Y. And H is equal to Z. Then come down, generation 3, Gen 3. L is equal to X. HB is equal to Y. And H is equal to Z. Do you understand? Okay, Alia has given an answer. I'm not asking generation 1, okay, but still you have given generation 1. Generation 1, okay, cool. That's very... Okay, so Gen 1. So you have given me, okay. Gen 2. Okay. Gen 3. Oh my God. So Alia has made a very, very beautiful mistake, much beautiful than this whole experiment. I'm not, that's why I asked you to give your answers in the chat. Others, please, quickly. Something very, very simple sometimes can be confusing, but we should not leave it. Okay, Iram has given an answer for generation 2, generation 3. Okay, but Iram, there is a problem. Generation 2 will have 4, total 4, but yours just add up to 2 only, right? You've written 1H, 1L, and for generation 3, HLLL. How many total DNA strands will be there in generation 3, first of all? 8, right? Right, everyone?
Yes, sir.
Okay, so Iram and Alia both tried, but the answer is not correct. Mariam and Rabia and Aisha. Anyone is still giving an answer? Should I explain it to you? Okay, let's do it. Let's say we start with parental generation and both the strands are heavy. Red is heavy, green is light. In generation 1, and this is if we are following fully conservative method. So we'll start with heavy, but so this was grown on heavy nitrogen. But remember, to do the experiment now, we have to shift it to normal nitrogen, because if you do not shift it, the experiment won't work. So now when you shift it in generation 1, from 1 now there will be 2 cells, but if it is fully conservative, 1 cell will have heavy, but the other DNA will be both light. So in the 2, 1 will be heavy, 1 will be light. You understand? Now going to generation 2. Now remember, this 1 is still conserved. So 1 will still stay heavy, but all other 3 will be light. You understand why?
No.
Yep.
So this was only what I wrote in the chart.
Okay. Let me see
No, I
again. So
mean to
you,
say that 1 will be heavy and the rest 3 will be light. That's why I wrote H, comma, L, L.
but you are saying generation 3 there, aren't you? I asked for
Oh,
generation.
so sorry.
Yeah, right. Remember? I asked for
Yeah,
generation
I
2
just
and
got
3.
confused. Generation.
Okay. Then your answer is correct. But then when we go to generation 3, remember, 1 will still stay conserved, fully conserved, because it's opening and closing to itself only. That is what the concept of fully conservation is. But since all other 7 will be light, because every time you only have normal nitrogen to make new DNA. So in generation 3, you will have 1 heavy, 7 light. In generation 2, 1 heavy, 3 light. In generation 1, 1 heavy, 1 light. And generation 0, which is parents, only heavy. So that is what will happen if it was fully conservative method. In none of the generations, you would have got any hybrid band. So the fact that if you do a thought experiment, and if this was true thing, this would have been the result. But since Messelson and Stahl got the result which is shown above, everyone understood this, people?
Yes.
Yes, sir.
But since Messelson and Stahl very clearly got a hybrid band right in generation 1, it proves that what should have happened? Then they scratched their head and they thought, okay, what happened? What happened? We started with heavy parents. Why all the progenies are hybrid in generation 1? Because both heavy strand must have opened, like Watson and Crick said, and two new light strains would have formed. Two new light chains would have formed, because now you have shifted it to normal nitrogen media. And this is the only way you can explain this result. And this also proves that DNA indeed replicates through semi-conservative method and not through fully conservative or any other method. Okay, makes sense, everyone?
So,
Yes, Iram.
so when you're talking about fully conservative method, then it should apply also for the light nitrogen. Why only the heavy
Yeah,
nitrogen?
it will also apply for the light nitrogen. So this light
If it
nitrogen,
applies for the light nitrogen, how are they able to replicate? They should be also intact together.
they should also be.
Intact together, right? As the
See,
red.
when I say intact together, it does not mean that they are not replicating. So by fully conservative method, I mean to say that the molecule is conserved fully. So to replicate, of course, it opens, right? So in generation, for generation 0 to generation 1 also, both will open, two new strands will form, but instead of attaching to an older strand, they will attach together and the older strand will attach together. That's called fully conservation. So whatever was mine stays mine. Okay. Try to understand it this way. I had one notebook. You had one notebook. You are copying from my notebook. Okay. But that kind of a copying is fully conservative copy. You open my notebook, copy from my pages, but then close my notebook and keep it, it stays mine. And you close your notebook and that stays yours. That's fully conservative copy. Do you understand?
Yes, okay.
But semi-conservative copy is you open my notebook and whatever is on the left hand side, you copy whatever is on the right hand side, paste your page in my notebook, take my page and put it in your notebook. That's semi-conservative. So my notebook is semi-conservative. Half of it is mine, half of it is yours. Do you understand,
Okay.
Hiram and
Yes.
everyone? But whether it is semi or whether it is fully, it has notebook has to open to copy. DNA also has to open to copy. But normally when we copy in day-to-day life, we do fully conservative copy. Your copy is completely yours, my copy is completely mine. So here also, if it was fully conservative, the light strand would have opened, two new strands would have formed, but the older strands would have closed together and the new strands would have closed together. And does not matter because from that point onwards, you are culturing them into normal nitrogen only. So two light strands, when they will open, they will form two another light strands. Right? Do you understand?
Yes,
So
sir.
green
Yeah.
will make more greens. Red will open every time, but every time it will open, it will only close with itself, fully conservative. So that's why there will only be one red strand that came from parent, and it will continue to be that one red strand for infinity of generations. And every new strand will be coming from the normal nitrogen. Make sense, everyone? Give me a hands up. This was a little convoluted concept, but if you understood this, things will become very easy from here. Iram understands. Alia? It was a good question, Iram. Good clarification. Mariam, Rabia, and Aisha? Okay, so I'm giving you a homework with the image which I was thinking I will teach you today, but since we are out of time in understanding replication, conservation, what you can do is you people can click. Can you all take a screenshot of the screen that is in front of you? This thing. Oh, by the way, so all of you have been added to a WhatsApp group? Yes or no?
Yes, sir.
I
So,
think all of you have been added to a WhatsApp group with me, right?
yes,
Anyone
so
who's not in that group? Ali is there, Iram is there. Anyone who's not in that group? Mariam Khatun is there, Rabia is there. So 4 people. What about Aisha? Aisha, you are also there in the group, right? Are you raising hand because you are there in the group, or you are raising hand because you are not in the group? So there's a WhatsApp group created, and it's very, very useful for me. Yes, you are there. Very good. So everyone is there. So just take a screenshot if you can. Otherwise, I will share the same thing on the group. And just try to just see what's happening in this picture and read NCERT and write down what pages you have to read. This is your homework. Write down, you have to read page number 104 to 107. 3 pages, just 3 pages, 104 to 107. Okay. And if you read this in the next class, we will finish replication in half an hour and we can go to transcription. If you don't read, then it will take time for me to explain you things again. So will you do homework, everyone? Perfect. So let me just tell you about the players that you have to focus on. Remember, see, this is the DNA, which, let's start from here. This was the DNA, which was double helix. The helix opens first, then the zip. So it's like a zip, which is helical. So you unwind first, then you open the zip, 2 strands open, and on both the strands, 2 new strands are formed. But the problem here is polarity. One strand is in different polarity, one strand is in different polarity, but opening can only happen in one direction. So because of which, one strand forms continuous and the other strand forms in fragments. And this is what you have to understand.